Hello, my name is Katherine Do, and welcome to the video for Module 3 of the Clinical Metaproteomics tutorial. In this video, we will cover peptide verification. Our previous videos covered the background of the clinical metaproteomics field, as well as the first two modules, database generation and discovery. If you haven't already, please watch these videos to gain a better understanding in preparation for this video. So, let's begin. This video will follow a similar agenda as in our previous videos. We will start with summarizing where we left off in the discovery module, as well as where we are in the clinical metaproteomics tutorial. Then, in the hands-on section, we will start the verification workflow, where we will verify that the peptides from Module 2 are indeed of microbial origin. Then, we will query those verified peptides to retrieve their protein accession numbers, which we will also call UniProt IDs. Lastly, we will generate a database of protein sequences associated with our verified peptides. That will set us up for the next module, quantification. Here is the recap of the discovery module. We used the reduced MetaNovo database from Module 1 and our sample MS datasets as inputs for SearchGUI PeptideShaker and MaxQuant to identify peptide and protein sequences via sequence database searching. We used two programs to increase the range of detectable peptides. After identifying and filtering peptides, we created one single list of unique microbial peptides. This list will be an input for PepQuery 2 in this video. Let's see where that puts us in context of the clinical metaproteomics tutorial. Out of the 5 modules that make up the clinical metaproteomics tutorial, we have covered modules 1 and 2, which are database generation and discovery respectively. The focus of this video is the verification module. We will use PepQuery 2 to verify confident identification of microbial peptides. This is a crucial quality control step due to the high abundance of human proteins, as we want to avoid misassignment of human peptides as being of microbial origin. This ensures the trustworthiness of our findings. Then, using Query Tabular, we will query those verified peptides for their associated proteins. Proteins are cataloged by unique identifiers called protein accession numbers, which we will also call UniProt IDs in this tutorial. The Query Tabular tool will give us these protein accession numbers, but we still need to retrieve their corresponding protein sequences. To do this, we will put these accession numbers into the UniProt tool, which will generate a database of protein sequences associated with our verified peptides. This verified database will help us reduce false positives when we quantify our peptides and proteins in the next module. Next is the hands-on section. For this video and the remaining videos for modules 4 and 5, I will skip over much of the setup since we use the same one for all modules. If you are unsure about whether you've imported everything correctly, I will show how to put in the input data and start the workflow on the next slide. Here, I encourage you to pause this video and try setting up on your own, including creating a new history and importing the data and workflow. Be sure to put the sample data sets into a collection to keep files organized. If you need help, please see the previous videos in the GTN tutorial and FAQs. Following setup and starting the workflow, I will discuss the workflow more in depth. Now I'm going to show how to put in the inputs and start the workflow. We're going to match the uploaded datasets to the input names listed. Then we are going to click Run Workflow. Next, we will discuss the inputs and main workflow steps. We will end on the final expected output for this module, which will set us up for Module 4, Quantification. From Module 2, we obtained a list of distinct microbial peptides. These have been filtered to exclude human and contaminant peptides and grouped to retain distinct confident microbial peptides. This list will go into PepQuery 2. We have also provided the same 4 example datasets that were analyzed by LC-MS-MS as MGF files. These datasets will also go into PepQuery 2. Also from Module 2, we will need the SearchGUI PeptideShaker and MaxQuant peptide reports. These reports have not been filtered for human or contaminant peptides or confidence. The relevant data we need from them are the peptide sequences and proteins that will allow us to accurately match the peptides verified by PepQuery 2 to their corresponding proteins. Next, let's discuss how we are going to verify peptides in this module. Uniquely, Module 3 has been split into 2 figures, Part A and B, due to the numerous steps involved. In these figures, the major steps and tools have been depicted. I will give a general overview before delving deeper into what tools and outputs are involved. Details about tool parameters can be found on the GTN tutorial page. First, Part A, shown on this slide, encompasses using PepQuery 2 for verification. The goal is to verify that the microbial peptides from Module 2 are indeed of microbial origin. We will search the list of 196 distinct peptides against the sample data and against human and contaminant sequences. Additionally, the Query Tabular tool is used twice in this module, once in Part A and again in Part B. In Part A, the Query Tabular tool will be used to select verified microbial peptides that were detected with confident PSMs. We will require the SearchGUI PeptideShaker and MaxQuant peptide reports from the Discovery module for this step. This leads into Part B, where we will use the verified peptides from Part A to generate a compact protein database that will be used in our quantification module. The top left box labeled Discovery module is a quick reminder that we use the peptide reports from Module 2 to verify that these peptides are indeed of microbial origin. The Query Tabular tool will be used a second time to extract the accession numbers of proteins associated with the PepQuery 2 verified peptides. These accession numbers can also be called UniProt IDs, as we will put them into the UniProt tool. We will use UniProt to generate a database of protein sequences associated with those accession numbers. Lastly, we will merge and filter the UniProt generated database with human and contaminant protein sequences, resulting in a compact, verified database that will be used for peptide quantification in the next module. That was an overview of the module. Now we will talk more in depth about the tools used. We will use PepQuery 2 to verify what distinct peptides from Module 2 are truly of microbial origin. While we have consistently selected and filtered for microbial peptides throughout our analysis, this verification remains a crucial quality control step due to the high abundance of human peptides compared to microbial peptides. We want to avoid misassignment of human peptides as being microbial, as this would result in false positives. You may notice that we are using the second iteration of PepQuery here. The PepQuery tool was developed as a peptide-centric search engine for tandem mass spectrometry data analysis to verify the quality of non-host sequences by assigning p-values corresponding to the confidence level in peptide detection. This tool allows users to search a list of peptide sequences against a proteomics database to verify high-confident PSMs while also examining peptide modification. This greatly reduces false discoveries in novel peptide identification. The second iteration of PepQuery that we are using here builds upon the initial software by providing a new tandem mass spectrometry spectrum indexing, which results in highly efficient targeted peptide identification. So we have our list of distinct peptides that we want to verify, the sample MS data to be used for identifying peptides, and a reference protein database of human and contaminant proteins. Novel peptides that match these reference sequences will be excluded, leaving us with a list of verified microbial peptides. The PepQuery outputs we are interested in are the PSM rank files. These files will provide the identification information for the input peptides, including peptide sequences, modifications, p-values, and confidence assessments. In our analysis, we want the verified peptide sequences with confident PSMs. These peptides will be put into Query Tabular along with a peptide protein dataset that we will create from the SearchGUI PeptideShaker and MaxQuant peptide reports. This is the first of two uses of the Query Tabular tool in this module. Not only do we need to verify that our peptides are of microbial origin, we will also need to make sure that these verified peptides are accurately assigned to their proteins. First, we will take the peptide reports from SearchGUI PeptideShaker and MaxQuant and create a new data set that contains two columns, peptide sequences and protein entries. Keep in mind that this is a different data set from the list of distinct peptides that will be verified by PepQuery 2. The Query Tabular tool will use the peptide protein dataset to accurately assign verified peptides to their proteins. So, at this point, we have obtained verified peptides and their corresponding proteins that are cataloged by their accession numbers. As I previously mentioned, these accession numbers can also be called UniProt IDs. In our second use of Query Tabular, we will extract the list of UniProt IDs. At the time of this video, we obtained about 73 UniProt IDs. These will be fed into the UniProt tool. The UniProt tool will download the protein sequences that are identified by the UniProt IDs we obtained. To summarize, we went from verifying microbial peptides to matching them to their proteins and getting the accession numbers, and now we've obtained their protein sequences. We have arrived at the last step of this module. We will take the UniProt generated database and merge and filter with human and contaminant proteins. This verified database will be used in the next module for peptide quantification. This slide is a bit of a tangent. You can skip this if you want, but I did want to show an example of how a protein sequence in the UniProt KB database is cataloged in FASTA format. I won't explain everything on this slide, but I do want to highlight some of the FASTA headers. First, the protein accession number is highlighted in green. Here we are showing A0A135ZGE9. Then, in yellow, is the recommended name for this protein, which is methionine aminopeptidase. A recommended protein name is unique and allows for unambiguous identification of a protein. Highlighted in blue, we can also see the scientific name of the organism. In this case, we have Lactobacillus crispatus. Overall, UniProt entries will follow the same general format. If you want more information, I've added a link to a UniProt page that explains the FASTA headers in more detail. This marks the end of Module 3, Verification of the Clinical Metaproteomics tutorial. To navigate to the next module, go to the bottom of the verification page until you reach the Go Further box. Click the link to get to Module 4, Quantification. Please feel free to share feedback about what we did well and how we can improve. We hope to see you in the next video where we will cover Module 4, Quantification.